Right, well, welcome everybody. My name is Laura Seth, here with Aptitude, Marketing Director. We wanted to welcome you to the Aptitude 2020 webinar series, which we will be doing a quick overview of who Aptitude is, and then I will be passing it on to our guest presenter. As we're going through the presentation today, please go ahead. If you have any questions, we will have time at the end for a question and answer session, so please go ahead and be thinking of questions for Troy. And as I mentioned earlier, if you could please go ahead and mute your lines, we would greatly appreciate it. So for those of you not familiar with us, Aptitude is an approach that many of the nation's larger, sophisticated purchasing organizations are leveraging to accelerate their supply chain activity. It's a process that is being adopted by value analysis teams, contract managers, supply chain professionals, and clinical staff. As a result, providers are more effectively managing their PPI strategy, accelerating value analysis structure, gaining line of sight into opportunities, and viewing market trends to better align with the right strategic partners. It's an all-encompassing approach. From cross-references to responding to bids, it's all streamlined into one platform, resulting in a simplified, more efficient contracting process. It also accelerates and delivers speed to contract. Standard T&Cs mean less time and legal, and again, the single platform creates efficiencies that result in 60% shorter contract time. And the third benefit for suppliers is business intelligence. They have analytics to create meaningful dialogue and transparency into contract performance. It creates a single source of truth for you and your suppliers to align your goals and have informed conversations. So you can see why a supplier would choose to do business through Aptitude and how it enables them to offer the additional value that organizations like yours are seeking. Suppliers are passing value to providers because they are seeing value through Aptitude. So now let's turn it over to today's content expert for today's webinar. Joining us today is Aptitude's own strategy director and physiologist, Troy Tuttle, to discuss implications for drug-coated balloons with paclitaxel. So welcome, Troy. And let me go ahead and pass the presentation over to you. And as a reminder, once again, everyone, if you could please mute your lines, we would greatly appreciate it. Got someone typing on here. Not sure who that is. Go ahead and share your screen, yeah.
Perfect. Thank you very much, Laura. And hello, everyone. Welcome to today's call. And as I get the presentation loaded here, we're excited that you're able to join us here on a Friday. I know that we could be doing a lot of things out there, and you've decided to join us, so that does not go unnoticed. And today it's really an intriguing topic. With paclitaxel and med device. So the spirit of today's call is to cover that in detail and go through some important points. So get your Erlenmeyer flask ready and your compass as we talk a little bit of science and also some guidance. And specifically on today's agenda, what we have teed up for you is a review of angioplasty and some of the remodeling that happens post-dilation. Looking at paclitaxel, its discovery and subsequent mechanism of action that has it applicable to today's conversation. Looking at drug elution and subsequent device development and deployment. And then lastly, looking at the recent findings and guidance. And as I've looked at the current literature and the reviews out there, what's happening with regards to communications and to next steps. Taking a look at angioplasty as a review, I think it's important to note, some of the fundamentals that are happening when we deploy a solution in the vasculature. So, of course, we're looking at addressing plaque or narrowing of the vessel, and how do we open up that passageway to allow for increased blood flow. And we're deploying balloon in this case to go ahead and open up the vessel and compress that plaque against the sides there. And I want you to note a little bit extra expansion of that vessel typically beyond its current normal state. And at the bottom here, once we have that vessel open, we have a lot of options. We can go ahead and just use a balloon, standard angioplasty, or if we did decide to use a drug-coated balloon, that would also be utilized, or we could deploy a stent in the lumen there to maintain patency and durability if we chose to do that. Taking a closer look at some of the dynamics that's going to happen in the vessel post-dilatation, it's important to note that once we get that expansion of the vessel, there is some elastic recoil that happens in the near term. But it's really what happens over the course of a longer time frame that can look at remodeling. And that's where we're activating the cellular structure in the different tissue layers and can get some hyperplasia. So with that, the cell is now rapidly dividing. We have high rates of mitosis, and this can actually circumvent and be antagonistic to what we just did to help the vessel and keep it open. And we can get restenosis or re-narrowing of that vessel. So tuck that away in the back of your mind right now. We're going to switch gears real quick over to paclitaxel and take a look at its discovery. Back in 1962, researchers were out in the forest, basically looking for natural products that might cure cancer. And they went ahead and collected samples of the Pacific yew tree. Well, they took those samples, and analysts in Triangle Park, North Carolina, discovered that extracts actually had cytotoxic activity. From 1965 to 70, we had studies looking at isolating those components, and in 1977, we had mouse models looking at the leading tumor types across the population. And Dr. Horowitz identified the mechanism of action on microtubules, and then production then did get scaled. In 1984, we got the phase 1 studies coming into play. In 1989, this was the pivotal study at Hopkins where they found patients with advanced ovarian cancer. If they gave them paclitaxel, we saw at least partial or complete responses in 30%. That was significant. And in the early 90s, we saw a number of indications for different cancer types showing progress in halting mitosis. And as we dive into a picture of the tree and the bark here, it gives you an idea of a person standing here of just the magnitude of these trees. And there was actually a pause point and so many being cultivated for drug that they needed to do a little bit of protection. And now we do have different chemical means to go ahead and still obtain that drug for the utilization. I think it's important now to look at the mechanism of action of paclitaxel and exactly why it is so effective in the cytotoxic process. And so here we're at metaphase, and if you remember back to biology, this is the part of the cell cycle where the chromosomes line up in the middle, and then it's important now that they migrate to each pole. And what's important in that migration, as you see this cell start to pinch apart, is the disassociation of the microtubules and basically breaking those down so that way that DNA and the chromosomes can appropriately migrate to the poles. And looking at that microtubule formation in a little bit more granular way, you can see from A to B how they build during the initial phases of the cell cycle, but we're concerned with how B transitions down to C, how those tubulin dimers come off and then subsequently let that microtubule shorten and then allow that chromosome and allow that DNA to subsequently move. And so if we look at normal mitosis, where we can go from metaphase to anaphase and have those chromosomes migrate, paclitaxel literally binds to the interior of that microtubule and stabilizes it. And it does not allow that DNA to migrate from the middle to its appropriate pole. And so what that does is the cell basically freezes in metaphase, and then the cell signal ultimately leads to apoptosis, where the cell realizes that it cannot divide, and so it go ahead and then it does go and program cell death. So knowing that and coming back to our earlier diagram and knowing that after some angioplasty and treatment that we can have cells that are rapidly dividing in this certain microenvironment, we can see that the deployment in the appropriate dose and with the appropriate method can go ahead and help maintain that vessel staying open. Now, as we look at deploying paclitaxel, some questions come up, right? How do we get this molecule, which happens to be hydrophobic, meaning it doesn't like water, how do we get it into the tissue? So we can coat a balloon, just like is done with the drug-coated balloons. We can use a polymer on a stent, and then we can also just, have it directly applied and have it elute off another stent that's out in the market. So some important points come into play here with regards to how we move the drug and get it into the tissue walls where we actually need it to be effective. And so we're going to look at some cross-sections of those tissue layers and talk about how that's going to happen. So again, we're looking at that pivotal point where the balloon or the other device meets up with the tissue to see how we're going to move it. So in this example, we've got paclitaxel, which is called an excipient. So this is a specific compound that blended with paclitaxel enables it to migrate through the plaque and into those tissue layers where we need it to be. There's a number of different excipients that are in the market. And as you can imagine, when you inflate that balloon and now move paclitaxel and the excipient against that wall, there is some loss in that expansion of that balloon. Also, as you can imagine, if you're entering, typically, as you're moving the balloon to the area, to the location where you want to treat, that just moving that balloon through the vasculature, you could also have a little bit of drug loss. Interestingly, when they coat the balloons, they typically do them halfway inflated, and that way when they deflate the balloon, you've got a lot of the drug staying in the folds and creases to ideally maintain as much as you can to the site for delivery. And in the literature, there's a number of different percentages of how much actually makes it into the wall and then how long it does stay there. And the form of the paclitaxel is also important. If it comes in crystal forms, it can actually stay embedded and have a presence in the tissue for a longer period of time. And so that's important with regards to efficacy and actual overall outcome. So I put together a timeline here with regards to some of the early devices that came into the market, some expanded indications, and then what has us here today, talking about the meta-analysis by Katsanos and the mortality signal that he found. And so we've had some really strong platforms in the market for a number of years, and they have dynamically changed the way within which patency has been maintained. From the literature I read, we're right around 20% of patients got restenosis after angioplasty, and now that number is, below 5. And so we're seeing just amazing durability with these devices and platforms, and then the expansion into different sizes and indications. And then, late last year, we had that study come out from Katsanos with that meta-analysis looking at some studies that were showing a mortality signal at 2 and 5 years. So nothing at 1 year, but all-cause mortality, showing an escalation at 2 and 5. And then subsequently, we've had governing bodies come in and issue letters and statements. And then, of course, here in June, we had the big meeting on the 19th and 20th. So over 2 days, we had panels, we had research, we had industry coming together to really discuss what was happening. And as we take a quick look at the Katsanos study in just a little more detail, I just wanted to highlight the evolution from 1, and then at 2 years, we were looking at 12 studies or about 2,300 patients. And then at 5 years, the number got down to 3 studies and 863 patients. So smaller sample sizes than we're typically seeing in your large randomized studies to get a mortality signal. And then lastly, just how this paper and conclusion ended. Urgently warranted was pretty strong vernacular at this time, and I know it caused kind of some polarizing reactions. I've had some facilities that put a full moratorium on the devices, and then I had most of them saying, hey, we're going to let our practitioners do what they do best, is assess the patient, get them the right treatment, and then subsequently go ahead and take them to where they need to be with regards to disease resolution. And reviewing all of this, there are some important insights based upon the panel and other researchers that have come out with opinions here. And I think one of the most important ones is there is consensus that these provide benefits with reducing restenosis and reinterventions. And again, preventing that vessel from coming together again, and then preventing the patient from having to go and get an intervention. There is just consensus that these devices are helping patients. Collectively, we know we need more data and expanded follow-up. And not really starting new trials, if you will, but looking at ongoing trials. And there's around 20 studies with about 10,000 or so patients that I identified that are looking to be improved in the way they're tracking follow-up. and wanting to look at mortality. So that's a good way within which we can move forward. I believe that there's likely going to be some form of labeling changes that just don't maybe talk about a possible mortality signal, but also bring out the benefits of it too. So I think they're going to do a good job of being both sides, having a message there. Some of the smaller trials that were leveraged in the Cassano study had an unusually low mortality rate in the angioplasty only arm, and this really helped diverge and get results, as I've learned, that weren't really expected. Better overall study structural designs are needed. This has collectively been a narrative out there with everyone is saying, hey, on the upfront, we need to make sure that we get the right endpoints established early. So these meta-analysis are looking at previously contemplated endpoints. And then, of course, lastly, the best way to assess mortality is to establish it as a primary endpoint at the start of the study. On and on, this has been determined as one of the best ways within which we can make sure that there are no issues with looking back, but that we've contemplated this going forward. The last slide I have for you today is collectively going through the 12 key points that were covered on June 19th and June 20th at the most recent panel. These first 5 were covered on day 1, and then 6 through 12 were covered on day 2. And on the mortality signal piece, there was alignment that there is something in the data. But the true caliber of that is just not clear due to data irregularities and that limited trial size that we did take a look at. As far as the class effect, there was the inability to confirm anything for late mortality. Data integrity, again, resonates. There's the gaps in the data to hinder an accurate analysis. There was some talk about looking at different subgroups within the data, but again, the caliber and the robustness of it is currently preventing that. Mortality cause, there was definitely the inability to find a clear mortality predictor. And then on dose response, looking at the size of the balloon, of course, can have an effect on how much drug is delivered to a patient. The data just didn't show that the larger balloons had a higher mortality effect. And then on the industry side, really saying that there was no relationship based on their interpretation of the data. Preclinical studies, there was talk about looking at different animal models to see what can be leveraged here for insight, but their overall value was really minimized and said, hey, let's look at the current studies and really reaffirm what's happening there. Benefit risk, this one was clear. The near-term benefits certainly outweigh the risks, and devices should remain on the market with regards to evolving them in the next steps. Post-marketing surveillance, more data and collection needing to be extended. So I think that's one thing that we know is even beyond 5 years, continuing to follow up these patients as much as we can to continue the data collection. Labeling, like we talked about, it'll be updated, and I believe that likely it'll have just benefits and possibly just an enhancement. And as I've talked to different physicians about this, their narrative with the patient, their talk track will be a little different. As far as changing the informed consent, I've not heard that would be front and foremost for them, but just that their identification of the patients and a more upfront conversation will likely happen. Study timelines, maintaining that 12-month study requirement for indication of these devices, but then having some enhanced follow-up post that. And then ongoing studies like we talked about. The Viva group and a number of other ones will be looking at current studies and how to optimize and continue to address this matter. And then for the SFA, expanding beyond that indication to seeing if there's going to be any signals coming in with regards to the other areas for treatment. So with that, I definitely appreciate your attention in learning a little bit more about where paclitaxel came from, how it made its way as an important drug for treatment. And then, next steps, we're ideally going to be having another letter and follow-up here in August from the governing bodies to direct us on where to go. So with that, Laura, I'll pass it over to you for any questions or feedback we might have.
Perfect. Thank you so much, Troy. I really appreciate all that. Yes, let's go ahead and open it up for questions. You're welcome to either unmute your lines and ask Troy directly, or you can use the chat function here on the WebEx. And don't be shy. All right, looks like I've got one here on the chat. Does balloon inflation time have an effect on the dose delivered?
Yeah, it's a good question. Typically, I'll see different inflation guidances, anywhere 60, 90, 180 seconds, and what the research is showing is that that's not having a huge effect. Usually what you're going to get at the 90 seconds versus the 180, it's quite similar, and that the outcome, depending on inflation time, that there's no difference. So I would say, definitely follow the indication guidelines for that particular balloon, as they do have some different doses of paclitaxel that are layered on. But systemically, that's going to be how the inflation time is going to be monitored for that particular aspect.
Okay. Anybody else on the phone? I think in general, we'd probably all would like to know more about what you're hearing, maybe some directly from the physicians. Any examples you can elaborate on?
Yeah, so in my conversations, it's really been we wish we could have been part of this at the beginning with regards to when the data hit, and instead of kind of being so reactionary, how we could have been part of reviewing that data and putting some more rigor into how this came to be. And that while some trials have stopped with regards to this, I would say that practice is going to be based on the patients. And when I hear physicians tell me that, hey, this patient wants to be able to go, pick up their grandchild, and they want to be able to go to their job, and they realize that if there is this little bit of risk, that the benefits certainly outweigh that. And so what I'm seeing is still a strong use of these devices because of how effective they are, and that I really see everything still moving towards clinicians having this tool in their tool belts because it is so important.
Wonderful. Thank you. We do have another question that came in on the chat. Is there any difference in the DCB balloons from various vendors?
I would say on the scope of that right now, that the differences between them have to do, there's going to be different sets of data, I would say, right, and it's the viewpoint on how you see that data. I think they all do a great job beyond the basic angioplasty approach, and while they may use a different excipient, they may have different inflation times, that categorically they've definitely improved the basic angioplasty durability of the vessel.
Great. That answers their question. Anybody else? Got a few more minutes here. All right. Well, if there's no other questions, then feel free, if you have anything directly you'd like to follow up with Troy. He is troy.tuttle@aptitude.com, or you can always send in any questions to ask@aptitude.com. We appreciate your time today, and we look forward to hearing or seeing you guys next month on our next 2020. Thank you so much.
Thanks, everybody.
Bye-bye.